If I may please introduce our third and final speaker, Jonathan Heeney, Professor of Comparative Pathology and Head of the Laboratory of Viral Zoonotics at the University of Cambridge. He received doctorates in both veterinary medicine and pathology in Canada before a PhD at the National Institute of Health in Maryland. And this is where he first worked on coronaviruses. He was then a postdoctoral fellow in human pathology at the Stanford School of Medicine before moving to Europe. He was an Isaac Newton Fellow from Trinity College in Cambridge. In 2017, he established DIOSynVax Ltd, a Cambridge University spin-off to develop innovative genomics and computational approaches to improve vaccines. His fundamental research at the University of Cambridge focuses on how viruses co-evolve with species and, like SARS coronavirus 2, jump from animals to humans to cause disease pandemics such as COVID-19. Now he will tell us about the application of genomics to the development of immunity and vaccine approaches for SARS-CoV-2.
Good afternoon, Lauren. Thank you very much. I've put the genome of this virus at the bottom of the screen and a structural image of it on top, because I'd like to talk about the most important part of the genome down here on the right-hand side in red. Now, this is one of the largest RNA genome viruses that we know about. It encodes up to 26 different proteins. The 4 on the right-hand side are the most important for vaccine targets. They include the spike, which many of you have heard about, the envelope, this little green cloverleaf ion pore channel up here, the membrane, and the nucleocapsid. Now, let's just get herd immunity out of the way. What are some of the misconceptions about herd immunity? Then I'll progress to gaps in our understanding of protective immunity. The immunopathology we see that accelerates individuals to an often fatal sequelae associated with this coronavirus infection, and then the challenges of making coronavirus vaccines. So herd immunity. We've heard about this on the news. We've heard about it from politicians. Some have thought that this might be the solution to the coronavirus epidemic, and it's just a question of time. So herd immunity, 60% are estimates that the newspapers have put out there for the public to think about. And I just want to raise concern about herd immunity with the following slides. There's been various different approaches that individuals have taken in their own way of dealing with this. This is one I came across this morning in the Common, out for my walk. And of course, we've seen these images with the warm weather of late. But I want to emphasize that despite the epidemic and the great death toll that we've seen, especially in Europe, that the seroprevalence is relatively low. And even if antibodies were indicative of immunity, and I want to emphasize that they're not indicative of immunity, often these antibodies to coronaviruses are relatively short-lived. And this is an observation made with some of the circulating so-called seasonal coronaviruses, which cause common colds. So natural herd immunity is right now relatively intangible and potentially of high risk. And I want to emphasize that in the coming slides. So what defines protective immunity, and why is this such an important question? Well, this is a cartoon extrapolated what we know about from other viral infections. Upon exposure, there's replication of the virus, and that can be identified through PCRs of nasal swabs or isolations from their respiratory tract, even bronchoalveolar lavages. What few people fail to realize is that the virus is also excreted in our enteric tract and possibly spread this way as well. So as people progress either with mild symptoms, moderate, or even severe symptoms, they develop an antibody response. And these are, of course, classically IgM followed by IgG. And these are found both in people who are asymptomatic, mild, and who progress to severe disease. And I just wanted to emphasize here that there's an important dichotomy in the divergence of what happens between individuals who eventually recover and people who progress to disease with disease severity. Now, for the purpose of my talk, I'm going to focus on one protein, the spike protein, and that's that one that I showed you on the outer edge of the virion. It's primarily important for binding to the ACE2 receptor on our respiratory cells and our airways. And the key business end of this protein is a so-called receptor binding domain. And this receptor binding domain is what, in fact, is the primary target of what we would hope a vaccine would induce. You can see that this is a relatively large protein. It's a trimer made of S1 and S2 subunits, which are important for facilitating fusion to the cell membrane, entry of the virus, and release of its payload. And this structure here is just showing you that small receptor binding domain with that area that needs to be targeted by neutralizing monoclonal antibodies. So these are antibodies that, if they hit this sweet spot, are able to block entry of the virion. So what about immunopathology associated with coronaviruses? These are those adverse immune responses that facilitate that pro or hyperinflammatory response that is associated with the complement cascade and often coagulopathies and small clots that affect not only the lungs and the heart, but also the kidneys. Now, as we age, there are differences in our lungs and there are changes that happen with time. And there is a shift in the nature of the type of immune response that we elicit, not just within the lungs, but within our bodies. And this is a phenomenon often coined as inflammation. So there is a difference in the nature of the immune response that some think is associated with this pro-inflammatory phenomenon. There is a dysregulation of cytokine production and undoubtedly serious tissue damage. Not to say that mild or moderate disease isn't associated with some tissue damage as well, but it doesn't have the fatal sequelae as it does in the elderly and those with comorbidities. So back to the virus. As it replicates within the host, and as the immune response gets in gear and starts to generate antibodies, we get antibodies to a bunch of different viral targets. Some of them, of course, are targeting the S protein or the spike protein, and they're indicated here. And some of them will be fortunate enough to bind to the receptor binding domain. But over time, as one goes through the decades of life, we become exposed to other coronavirus, which may prime us to other antibodies that may not target necessarily what we need to target, which is the ACE2 receptor binding region that would block entry of this virus. So it's this decoration around the S protein that could lead to problems in immunopathology. And to illustrate, I'm going to take this cartoon published, I would say, in the night 2014, 2015, about feline infectious peritonitis, a domestic cat viruses which also causes a vasculitis and more severe disease. And the immunopathology is associated with two different potential putative pathways. One is called antibody-dependent enhancement, where non-neutralizing low-affinity antibodies will actually bind the virus and drag it into these myeloid cells that we have, for instance, in our lungs. The other is activation of complement via antigen-antibody complex, leading to vasculitis and edema, part of the sequelae that we see in SARS-CoV-2 immunopathology. So what about the challenges of making coronavirus in the absence of really knowing what the correct immune responses are and what we need to do in order to make a safe vaccine? Well, the pandemic has necessitated the acceleration of vaccine candidates that have skipped really the discovery and preclinical stage in animal models right into phase 1 clinical trials. And as we hear in the news, there are many phase 1 and soon to start phase 2 trials, a variety of different vaccines that are based on that full-length S protein. And this is one of the concerns that is thought to be potentially a problem, and which was demonstrated to be a problem with SARS vaccines when they were developed after the 2002-2003 outbreak. So what does DIOSynVax do differently than take an off-the-shelf S protein and put it into some sort of delivery vehicle? Well, what it does is it compiles the genomes from SARS-CoV-2, much like what Mike and Dr. Stevenson were talking about, aligning all those genomes together, looking at the similarities and differences. And what we focus on are really those conserved elements that are important and importantly related to structural proteins that need to be conserved. And those are what we focus in our vaccine designs. And we do this using computational biology based on known structures, synthetic gene technology to develop vaccine inserts. We screen these and develop candidates for clinical trials. To provide breadth, we also look at those other bat viruses, those that Professor Warby was talking about, that have led to the spillover not only of SARS-1 in 2002-2003, but that have led to the current epidemic and pandemic caused by SARS-CoV-2. We also hope to avoid any of the potential off-target effects that may induce immunopathologies such as immune activation. And we have a technology that is eminently scalable and versatile in that these inserts can be put into different viral vectors. And we achieve this, again, as I mentioned, by alignment of genomes that not just cover SARS-2, but also SARS-1 and the related bat sarbecoviruses. We use machine learning to help us generate the best T-cell epitopes to other viral proteins as well to include in these vaccines. So through this process of generating gene libraries and screening them, we're able to take out those vaccine inserts or those modified vaccine proteins that could potentially cause off-target effects. We take the best in class that are the most immunogenic forward, and we scale them up for clinical trials. So it's a proven pipeline that we have used successfully for the filoviruses. The filoviruses are a family of viruses that includes Ebola, Marburg, Bundibugyo, many other hemorrhagic fever-causing viruses. We've designed vaccines for arenaviruses, including Lassa fever and all its different lineages. And we're currently generating influenza viruses or vaccines for all the different potential influenza viruses that could cause pandemics in the future. So we're using this technology to generate coronavirus vaccines that are devoid of potentially immunopathological responses or inducing those immunopathological responses. And we're scaling these up for phase 1 clinical trials. Most importantly, we're trying to identify what those immune correlates or protective correlates could be and to develop diagnostic tests that would tell us really who may be protective in the future. Thank you very much.